Thank you, sir. Thank you very much. Thank you. We also want to felicitate the Honorable Minister with the memento of so thank you very much, sir. So we conclude the inauguration session and we will break for the coffee session, and after coffee session we will start with
our vote of thanks to Honorable Nitin Gadkari ji for having given us a completely holistic viewpoint which can be run in tandem with the policies of the government of India and suggestions which are going to be a challenge to all of us who belong in the scientific community. I am certain that the Vasantrao Dada Sugar Institute and all the scientists who are present here today, along with all the international delegates, will definitely take the initiative to work and plan on the suggestions that Honorable Minister Ji has given us. I thank you again, sir, for a comprehensive, holistic guidance which you have given our conference. Thank you again.
we now conclude the inauguration session, and after the coffee break we will start with the technical sessions. After a quick coffee break, we will start the technical session. The agricultural session will be conducted in the same auditorium, and the technical session will be conducted in the auditorium on the first floor. So kindly have your coffee and please come back within 20 minutes. So we have a coffee break of only 20 minutes. Thank you very much.
हॅलो चेक येतो का? हॅलो चेक माईक टेस्ट चेक. हॅलो 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 माईक टेस्ट चेक.
रेपाळे साहेब आपलं डॉक्टर रेपाळे
येतोय का? हॅलो चेक माईक चेक.
या. नमस्ते नमस्ते. Can we start now? Okay.
Hello. Good afternoon, ladies and gentlemen. We will start now the first session of agriculture. It is my honor and privilege to welcome you all to the International Conference on Sustainability, Challenges and Opportunities in Global Sugar Industry, which is hosted by the esteemed Vasantdada Sugar Institute. We are gathered here today to share knowledge, insight, and advancement in the field of sugarcane improvement. In this first session of agriculture on perspective in sugarcane improvement, in this first session of agriculture on perspective in sugarcane improvement, we have the chairman, Dr. S.R. Gadak, Vice Chancellor, Dr. PDKV Akola from Maharashtra, and I warmly welcome him on behalf of Vasantdada Sugar Institute. As we dwell in the enriching experience, let me take a moment to acknowledge the presence of some distinguished scientists who have contributed significantly to the field of sugarcane improvement. We are privileged to have with us today 5 remarkable delegates who have made outstanding contribution in the field of sugarcane improvement. I welcome today's distinguished speaker on the dais. From my right, Dr. Prakash Lakshman, Professor, Director, Sugarcane Research Institute, Nanning from China. Dr. German Serino, Director, Cacra Experimental Agricola from Argentina. Third one, Padma Shri, Dr. Bakshiram Sir, former Director, Sugarcane Breeding Institute, Coimbatore. Dr. G. Hemaprabha Madam, Director, ICR Sugarcane Breeding Institute, Coimbatore. And Dr. Michael Butterfield, Data Science and Scientific Affairs Manager, CTC from Brazil. I welcome all the honorable speakers on behalf of Vasantdada Sugar Institute, Pune. वो डिलीट किया हुआ है क्या? आपका दूं ना? नहीं, ये बीच में का, ये नीचे, ऊपर ऊपर ओपन. अरे गड़ा सर का
Dr. Repale, you can start now.
Yeah. Dr. Gadak sir is presently a Vice Chancellor at Dr. PDKV Akola Agriculture University. His specialization is breeding, development and release of new crop varieties. He had released varieties from sorghum, sweet sorghum, Niger, Dolices bean, cucumber and brinjal. He has a vast experience of 39 years released 41 varieties during his tenure as a director of research at MPKV Rahuri. He had made 267 recommendations and he had got awards, international one and other 9 awards, and with 123 publications. He had wrote 8 books he had guided 14 PhD students and 8 MSc students. With this, I hand over to the chairman to proceed the session.
Good afternoon to all of you. Friends, today we gather here the third international conference organized by Vasantdada Sugar Institute, Pune, on sustainability challenges, opportunity in global sugar industry. And now we are going to start technical session first. Theme of the technical session is perspectives in the sugarcane improvement. And on this occasion, the coordinator of this session, Dr. J.M. Repale, reporters, R.M. Davrutmat, and five speakers from the different places of the world. That is Dr. Prakash Lakshmanan, Dr. German Sirino, Padma Shri Dr. Bakshi Ram, Dr. G. Hem Prabha, Dr. Michael Butterfield. They will present their presentations here, and I request to all speakers only 20 minutes for presentation. You should complete your presentation. It is requested to all speakers to complete our presentation within 20 minutes, and be stick up for the time. We want to complete our session within stipulated time. And now this is a very important session of this now, and I request to the suggest to the coordinator, Dr. Repale, to go ahead one by one. Now, first speaker
First
is
speaker is Dr. Prakash Lakshman. Before
Prakash
he
Lakshmanan.
start his presentation, I will brief about his work. Next slide. Second slide. Dr. Prakash Lakshman is a professor and director at Sugarcane Research Institute from China, Nanning. The field of specialization is sugarcane improvement. It's a multidisciplinary approach. He had got awards, a recipient multiple awards for R&D excellence from Australian government, Australian Academy of Sciences, and Australian sugar industry. He honored as an editorial boards of several international journals. As an honorary professor at University of Queensland, Australia. He had quite 35 years experience in the field of sugarcane improvement and genetic improvement of sugarcane. He had around 200 plus publication in international and scientific journal. I request Dr. Prakash Lakshman to deliberate on his topics, sustainable sugarcane feedback production, challenges and opportunities. Dr. Lakshman, please. Next. It's your only?
Yeah, it is more. Thank you, Dr. Ripale, and thank you, the chairman, my distinguished and all the delegates, dignitaries. Now, let me first start with thanking VSI management organizing this conference for inviting me to speak on my perspectives on the an important theme that supply of green sugarcane on a long-term basis to meet the growing demand for bioenergy and sugar. So I think this morning you all heard from the keynote speaker and this afternoon from the minister, how important sugarcane is becoming in many countries, including here in China, in India. So let me just start this talk with taking you through some of the global statistics as well as the country-wide statistics to get into the theme of sustainable production of sugarcane for the energy requirements or for the renewable energy market. So this is the trend of sugarcane production globally for the last 6 decades. It was quite impressive. It increased quite reasonably well, or in fact quite impressively, until a decade ago, and then it started flattening off. That is what the data shows. From 2010 onwards, the average production, the global sugarcane production is somewhat staying at the same level, going a little bit up and down, which is mainly dictated by the weather events. Now, even this is the global trend. What is more interesting is, if you break it down, this global mean into country-wide performance, most of the production of sugarcane is happening in 3 or 4 countries. So basically, Brazil, India, and China account for almost 65% of the total sugarcane production. In fact, if you add Thailand, it becomes about 70%. So from a production perspective, global sugarcane production perspective, actually, it is all happening within 4 countries, these 4 major producers, Brazil, India, China, and Thailand. And anything happening in these countries are going to seriously affect the global supply of sugarcane and eventually the sugar as well. So they say that's the situation. And more significantly, what you need to look at here is, in all these 4 countries, the whole production, the 70% of the sugarcane production is done by just 8 varieties. 3 varieties in Brazil, almost half of the Brazil. It's a massive, 10 million production, and 10 million hectares. And then 2 each from India and China, and 1 in Thailand. So effectively, this 8 varieties produces bulk of the sugarcane in this world. That itself is a very risky thing because it is from a biosecurity perspective. If something happened to one of these major varieties, the production can be really crashed down. And that happened in some countries, even at least in Australia, some time back. So some of these risks need to be managed if we really want to increase the production in globally. Now, where are the demand for green cane coming from now? It's essentially the 3 reasons for the renewed impetus producing more sugarcane. One is the growing expansion, the rapid expansion of renewable energy market, which is really very seriously, you can feel it in India. And to some extent also you can see it in Thailand, some Southeast Asian countries, and of course in Brazil. So there is really a big push for renewable energies production from sugarcane. Then at a respectable level, the global demand for sugar is growing, even though per capita consumption is slightly declining in the last several years. So it is mainly the industrial demand for sugar is growing. So the sugar market is still really creeping up. Then finally, from a political perspective, I think all the major countries are really pushing hard for decarbonization, and sugarcane is considered to be one of the green crop or green bioenergy crop, and that is also pushing hard on sugar industries to make it more sugarcane production. Now let us take these demand side one by one, a little more, get into a bit more detail. The first is the demand for sugar, as I said earlier. So this projection is coming from S&P Global Commodity, outlook. They are a very highly respected, consulting firm, and they made this projection. And according to their projection, the demand will continue to grow at a reasonable level going forward next 10 to 15 years. And the assumption is again based on the need for sugarcane for the bioenergy sector. In India, it's really, really very evident from this afternoon, the presentation is more than clear that it is going to be, the government is going to make all efforts to adjust the policy decisions to make it. Here it is energy independence is a national objective, and government is making now E20 as a mandated criteria for fuel from 2025 to 2026, which is a very ambitious target. I'm not sure whether India can produce that much of sugar cane to reach E20 by 2025. But, well, that is, you need to be very ambitious in all these things to get somewhere. Then transportation fuel demand is going to go up almost. This is projected to make it more than double in the by 2014, because there is going to be an explosion of new automobiles in India. It is going to be predicted to make about 300 million new vehicles by 2040. So these numbers are really underpinning the push behind the policies set by the government to make the Indian sugar industry produce more sugar as well as ethanol. Then what are the options? So, well, there is a market force behind the need for more sugarcane production. But what are the options? How realistic it is to produce a substantial amount of sugarcane to make a dent on the energy market. Energy market is huge. I mean, just take the case of Indian fuel energy market. It's a huge one. If you want to make even some impact on it, the amount of sugarcane need to produce is huge. So where is these sugarcane is going to come from? That is one important question we need to ask. It can be, there are only three options. The first is increasing the production area. The second is increasing the productivity, that is yield increase, cane yield or sugar yield or both, and as well as making it more efficient in production on a sustainable way. So these are only the three options. The efficiency and sustainability is coming into fore because the decarbonization target of industry, government pushing for that element of production, agricultural production. Just take the case of production area. This, the left side panel, the left panel graph is showing the area increase, the production area increase in sugarcane at a global scale. It just like the production, it increased over the last 6 decades very well, and again plateau. And if you break it down, the country-wide increase, the right side panel give you the details about the country-wide expansion of sugar production. Actually, most of the increase in sugar production is accounted for by the expansion of sugar area in two countries, the Brazil and India. Most other major producers, so-called major producers, really the area of production didn't much happen. So essentially, the production increase is explained by the really rapid increase in the production area in Brazil. And that is perhaps the underlying reason for making Brazil so successful in bioenergy production. And in India, it was gradually increasing over the last 3-4 decades. However, the third producer, the biggest producer, China, it went the other way. In fact, in the last decade, we lost almost 1/3 of the Chinese sugarcane production. It was about 1.8 million hectares in 2011, and now it is about 1.1 hectares. A substantial amount of production area actually declined. There is a reason for that. The most of the lost area has gone for higher value agricultural production, mainly for citrus production and vegetable production. So in China, the Chinese sugar industry actually shrunk by 1/3 in the last 10 years. So when you look at the trend in most of the countries, barring these two major producers, and what happened in China, the prospect of expansion of sugar area to increase the sugar production is possibly minimal. Now, let us check the case for sugar yield. Like the area, the production increased. It's quite impressively around 315% over the last 6 decades. But the cane yield improvement was only about 40% during that period, which is not that impressive. But it is still a reasonable rate of increase in sugar production, sugar yield improvement, genetic yield improvement. Well, that also, like what happened in the production area, again, the production rate, the biggest improvements happened in Brazil and India. Now, I just mentioned earlier that the rate of improvement of sugarcane is relatively around just 40% over the 6 decades period. But when you compare that rate of increase, even though it is quite decent for a crop of sugar, the crop with an annual cycle and vegetative crop, when you compare it with other major crops, particularly cereals, and it's really quite low. Even sugar beet in the last 25 years made a very impressive improvement in production. It's increased by 160% over the last six years. So considering the improvement of rate of genetic improvement of other crops, sugarcane is actually not doing really well. So if that is the trend, can we expect to increase the sugar production to such a large area required for bioenergy production by relying on increasing the cane yield, which I doubt probably may not be a very wise assumption. However, when you consider the productivity gain on a countrywide in the last 6 decades, except the front runners, United States and Australia, where the productivity was actually reached quite high in the 80 to 90 or even 100 tons per hectare by 1960 and stayed on as it is even now, somewhat in the same range. But the other major players really picked up fast over the last 4 decades, in the 1960s by 2000, but again reached to this level and staying on. This is the trend which is worrying me now. If this is the case, everyone is going to stay on the same place, will the yield improvement is a viable option to make a quantum leap in sugarcane production for the market demand going forward for the next 5 or 10 or 15 years? So if you look at the next slide, breaking it down into the productivity trend of sugarcane yield in different countries, for example, Australia, Brazil, China, India, and Thailand, it's all staying flat. Remember, during this period, hundreds of varieties were released, and still it really didn't make any difference. So can we expect a real big quantum jump in productivity in the next 10 years if this is the historic trend of cane yield. And if you look at the sugar production, sugar yield, it is worse. It stayed on for a long time, somewhat in the like 13, 14% on stayed on. It really didn't change much. However, then one would wonder that, is there any possibility at all to make a big change? Is the varieties which we produce now already achieved the maximum productivity yield ceiling, and that is it, what we are going to get? But the data, some of the data, which is I'm showing in this slide, is proving otherwise. It is not actually true. For example, here in India, 40238, which is considered to be a wonder cane, or one of the very rare exception, which produced up to 327 million tons per hectare in a commercial setting. And multiple varieties produce around 250 tons per hectare here in Maharashtra and also in Queensland in Australia. So there are the same varieties which we are now currently growing. Some of them really produce very high yield in some farms under commercial settings. That is suggesting that it is actually, the yield potential is still very high, and there is something beyond the genetic block is really stopping the productivity gain achieving in a commercial production. So similar high yielding examples are existing in China, Thailand, and in fact, the theoretical estimation of sugar yield for sugarcane is estimated to be around 380 tons per hectare. So if all the production conditions are ideal, with no water stress restrictions or diseases or anything, you can produce up to 381 tons per hectare with the existing genetics. That means these data is telling that if we continue to grow or continue to produce varieties and grow them the way which we are doing, really we can't expect a big quantum shift in productivity in the coming future. So we may need to do something more differently. Just by doing the same thing again and again will not take us going forward. The current system of selection, just do the trials, select for yield or select for sugar alone, or fiber or some other traits alone is not going to take us far. We need to think a little more differently. Something like what the grain industry is doing. We may need to go for more production constrained, breeding for overcoming the production constraints in the farming systems. So that is probably one of the way the breeding program need to look at it. So what is expected in the future is, and what is currently limiting the productivities are the extreme weather events and also really worsening the land degradation, like soil acidification, in the weather front, drought, lethal heat stress, flooding, etc. So unless we really started thinking along that line how the grain breeding programs are heading towards, and which they are making real big strides in genetic improvement, which we have seen the percentages here in my previous slide. So we probably, from a sugarcane breeding perspective, we may have to think a bit more differently in terms of targeting breedings and selection. I will just take you through two examples before finishing this presentation. The first is a climate change or weather extreme related situation, drought, which is becoming more and more regular. I'm just showing you data, the projections for India. India and Pakistan are considered to be the two first countries going to be most severely affected by the climate change effect. And what is projected is the extreme drought events are expected to increase quite significantly, even a very short term, by 2030 in India. So are we really gearing towards, our breeding programs are really gearing towards making varieties more resilient to the future weather patterns or future climatic conditions? Another example in the case of land degradation. Here I'm drawing an example from China. So this is the change in soil, just taking a one parameter, change in soil pH in 1980 versus 2023, so I think 2020, within the last 40 years, so much of land has been, the pH, soil pH has been changed more towards acidic, almost about 2 points acidic. In currently, nearly 60% of the farm soil, nearly 50% of the farms with the soil having 3 to 4 pH. So severe acidity is a major problem, limiting the overall productivity of the Chinese production area in Guangxi. Why? Because intensive farming, monoculture, there is no fallow period, too much of fertilizer, nitrogen fertilizer. The average is about 600 kilograms per hectare, which ranging from 345 to 1,300 kilogram nitrogen per hectare. So effectively, we are putting too much of input to destroy the production. So unless we are really prepared to tackle the cases, tackle the future scenarios going to be witnessed by the sugarcane industry, it's going to be very, very hard. We have a few more minutes. Do we have a few more? Okay. So I'll just take what we could do to tackle these problems just by showing one example of making varieties drought resilient. This example, I'm drawing it from the Australian breeding program, where I was involved in at that time. Improving drought tolerance and water use efficiency in sugarcane. So using the actual breeding materials coming from the Australian breeding program. So we initiated a program about 12, 14 years ago, a major program on identifying traits potentially suitable for using as a selection target for variety as clone selection in the breeding. So started with a massive selection program using the genetic material available at that time, including parental lines and breeding clients. The trouble is, when you look at the drought tolerance literature, there are nearly 100 different traits were published as excellent traits for improving drought tolerance. But when we selected some of them and started investigating in the field condition, most of them are useless, absolutely useless for using as a selection criteria for actual breeding program. Why? Because most of the traits don't fit into these minimum characteristics needed to be successful for a breeding program. One is the traits which we are going to select for drought tolerance or any other tolerance limit, the abiotic stress tolerance. They had to have very high genetic variation, must be highly heritable, and in fact the heritability must be more heritable. It should be more heritable than the yield itself, and must be good at diverse production conditions. There shouldn't be any yield penalties if you select for that trait, and it should be allowed out of season selection. So, and must be quick, easy, and inexpensive. So unless you feel these traits, it is really very hard. If a trait is not meeting these criteria, it has very little value for using it as a selection trait. Finally, we ended up with two traits, canopy conductance and the canopy temperature. Those two traits proved to be useful for using in the real breeding selection condition as the traits for identifying clones better resilient to water deficit and water insufficiency. So there will be huge genetic variability for the material we screened and for the canopy conductance, and that canopy conductance was very closely related with the photosynthesis. So that means the biomass production. It was very closely genetically correlated with the biomass production, and which was under both drought and irrigated and semi-irrigated condition showed a very high genetic correlation as well. So all these, the characteristics are turned out to be, these two traits turned out to be a very suitable fit for selection. And finally, using the aerial phenomics, we could actually easily screen large populations of breeding material, breeding trials, breeding clones, tens of thousands of breeding clones in a relatively short period of time, and showed very huge, using this phenomic system, and it showed very large variation. We can very easily pick up all the variation for
Dr.
these two
Prakash,
traits.
restrict to time.
Two traits, and it is now it is being implemented in the breeding program and currently undergoing in actual selection program in Australia. So with that, I will stop. Thank you.
Thank you, Dr. Prakash Lakshman, sir, for your thought-provoking and presentation and sharing your unparalleled expertise with us today. As decided, we will discuss after 5 presentations. It is my distinct pleasure to introduce our next speaker, Dr. German Serino, Director, Carca Experimental from Argentina. He is a Director, Carca Experimental Research Center from Argentina. His field of specialization is plant biotechnology and molecular genetics. He has released the first field trial of GM sorghum in Argentina in 2005. After that, he integrated the team that released and conducted the first field trial of GM sugarcane in Argentina in 2005. He had a vast experience of 33 years plus, and on his name, 2 patents are there. And presently, he has operated the genetic improvement of sugarcane in Argentina. He had awarded different 4 awards with 38 plus publication in international scientific journals. He has ICSB representative in the meetings from 2004 to 2007. So I request Dr. German Serino to present on his topic, genetically engineering sugarcane at Carca Experimental Agricola Rosa, from the research lab to the field. Dr. German Serino, please.
Thank you very much, and thanks again for the opportunity to share some of my work with you. So basically, I was asked to speak about transgenic sugarcane, and I would like to share our view about what to do with a transgenic sugarcane today. And of course, this is from our perspective, which is the small/mid-sized genetic improvement organization. So I'll start by introducing NA05860. This is the most planted sugarcane cultivar today in northern Argentina. As every other commercial sugarcane cultivar, NA05860 ultimately derives from a cross between 2 different species. One of them being Saccharum officinarum, noble canes, native originally from New Guinea, and this is Lahaina officinarum in the picture, from a picture of herbaria in Hawaii. And also to a plant that you know in India, which is Saccharum spontaneum. It's also called cans here in India. It's a wild species. It's distributed throughout Southeast Asia. So this process of repeatedly crossing noble canes with wild canes was called nobilization. It's a recurring series of crosses between spontaneum and officinarum, with officinarum being the recurrent parent. This picture from Angelique Don's laboratory shows you how officinarum and another species, Saccharum robustum, are actually the same species, whereas a wider range of genes are observed and markers are observed in Saccharum spontaneum in orange in the picture. So any hybrid would have to be more or less within the area between these 2 species, like the ones that are marked as outliers in the square. So also from Angelique Don's lab is showing that saccharum hybrids, this image showing that saccharum hybrids are polyploid, aneuploid, and their genomes are large and complex. And I'm showing you this because it has to do with what I'm going to show you down the road in this presentation. And I start by this. Because of this genetic complexity and also because of some characteristics of the crop itself, cane breeding, as represented in this flow chart from Chakra's breeding perspective, is labor and resource intensive. There are many decisions to be taken, many measurements, and a lot of work. So luckily, sexual propagation is only used for breeding and is not used for commercial propagation as other crops as corn, soybeans. So sugarcane transformation happened in 1992 in Robert Birch's lab. He enabled then efforts of genetically engineering sugarcane from there on. The technology that he developed more or less is the basis for everything we did down the road, even if there are some modifications that included agrobacterium, transformation, electroporation protocols, and so on. So I'm going to show you a few examples of what's being done, interestingly, from the application perspective in different work groups. In South Africa, Sandy Snyman's laboratory in Sasri genetically engineered sugarcane resistant to the glufosinate herbicide and showed useful characteristics in field trials. This has also been done by other people in the case of glyphosate resistance. Here I'm showing you Nasir's group work in Turkey with glyphosate-tolerant sugarcane. In Brazil, there's an interesting case, a little bit about this in the coming slides. There's a BT sugarcane you probably know about. It's resistant to Diatria saccharalis, the main insect plague in Brazil, and fairly reasonable damage control in BT sugarcane against the same variety with no BT gene. Also, Robert Birch's lab had shown a case of disease resistance, in this case, leaf scald. This is really far away from commercial, though. And also far away from commercial are some, this is contradictory, but anyway, resistance to abiotic, drought, and saline stress, which has been tested in several places. In Brazil, they also demonstrated that sugar is free of DNA and protein traces. This may not have been any news for many of us, but it is important from a regulatory perspective. And in this chart, you can see where in the extraction process for sugar you get rid of DNA and where you get rid of proteins as well. And so genetically modified sugarcane was approved in several countries. So I'm going to go by time. Basically, here in Indonesia, the first transgenic sugarcane released from 2011 to 2013, with some approval for feed in 2018. And these were done with a glycine betaine gene, which is supposed to protect against drought stress tolerance. There's not much publications about this regarding the effectiveness of this trait, but here it is. In Argentina, Estación Experimental Agroindustrial Obispo Colombres has transformed also sugarcane with the glyphosate tolerance gene. It had approval for food and feed, but has no approval for growth, because the Argentine industry thought they were not ready for commercial sugarcane at the point they were trying to release this, which was 2015. So, and then in Brazil from 2017 to currently, There's been a series of releases of Cry1AB, BT gene in different varieties from CTC. They have obtained different sorts of approvals, and this cane is being grown in the field. And the forecast for 2022/23 was 70,000 hectares to be planted with this variety. I have no record of this being accomplished and no forecast for 23/24 year. But anyway, this is what we're standing today. So where do we go now with this news? So in Chakra, just to tell you a little bit about us, is a nonprofit organization. We're focused on sugarcane variety development for the provinces of Jujuy and Salta, and this is the northern tip of Argentina. So basically in this pie, representing a little bit more than our sustaining area, and by sustaining I mean mills that pay for our research efforts. And our varieties are adopted as of 2022 in about 62% of the hectares. This is increasing since at least 2020 and earlier on as well. And we believe we may go up in the next coming years. So Chakra is kind of gained some reputation because of the adoption of the NA 5679 cultivar back years ago in South America, particularly in Brazil. So it was one of the most planted cultivars. NA varieties are demanded as parental genotypes in breeding programs international. So we're quite active on variety exchange programs, germplasm exchange programs, I mean. So initially in Chakra, we started our genetic engineering program, focusing as one of the main traits, virus resistance. This is the case for mosaic. We did a reasonable amount of work regarding identifying mosaic virus populations, generating transgenic constructs for resistance to mosaic, and generated a lot of transgenic events, 400 events, putatively resistant to mosaic, and they were tested in field trials. And we came up with something interestingly resistant, which after 12 years of the trials kept resistance. And these were 3 GM events. This is done through kind of very well-known technology called RNA interference or RNA silencing. And this is done through a mechanism looking more or less what I'm showing you here in this cartoon. But I'm going forward to the next trait that we're working with, which is glyphosate tolerance. We developed glyphosate tolerance in Chakra back in 2000s, and the field performance and trait efficacy assessments identified one event that sustained its performance during multi-environment trials. This is for cutting a long story short. We started building a regulatory dossier addressing biosafety issues, including trait performance, stability, witness potential, traceability, and we're submitting our dossier by early 2024 in our new attempt to release a transgenic variety in Argentina. Through this process, we have gained some engineering, trait development, and regulatory science know-how that we think will facilitate developing further GM events from new cultivar donors, constituting a sustained and current biotechnology product pipeline. The acquired know-how and future lines of work in transgenic sugarcane at Chakra will involve developing insect-resistant constructs via multiple mode of actions, and this will be stacked to herbicide resistance. We'll optimize our product design. We have learned that early molecular characterization of GM events may facilitate and ease some headaches that we had down the road in the previous efforts. And we think will notably reduce our regulatory timeframe based upon the experience that we gather, both in trade development, dossier building, and also by developing analytical methodologies. So we're also playing around with genome editing. We had some work published in the last, will be published, sorry, in the next coming issues of Sugar Journal. We're looking at herbicide resistance as well. In this case, has gene-based herbicide resistance for imidazolinone, sulfonylureas, and other herbicide types. This requires some fine-tuning of homology-directed repair to achieve functional knock-ins in the ALS genes. So far, what we've managed to do is mess with the gene in the right place, but we haven't got the modifications that we needed. We need to fix this. We need to find new DNA-free transformation protocols and also selection of transformed plants for this herbicide resistance. It's a lot of work, but we're there. Other lines of biotechnology work are easy. They're kind of byproducts of what we've learned so far. One of the things that we're doing is doing a new cultivar improvement based on some clonal variation. So we know that many varieties increase their stock number after transformation, and it's not because of the transformation per se, but because of the somaclonal variation that occurs upon embryogenic route of development. Like this, you can increase stock number. You won't increase the total cane yield, but you will improve a lot of interesting traits together with the stock number. We're applying this to some varieties. Some varieties, it doesn't work well at all. We're also applying a massive in vitro propagation facility targeting, at this point, about 2 million of high-quality clean cane seed per year. So as for conclusions, our future developments in sugarcane genetic engineering will be shaped by biotechnology as well as industrial and market developments in the industry. Because these developments occur at a rather fast speed, implementing adequate technologies based upon accurate anticipation of industry scenarios will define the success of each sugarcane research strategy. Before I go on and thank you, I wish to invite those of you involved in pathology and entomology to the next pathology and entomology workshop of the ISSCT, which will be held in Salta, Argentina. I promise you it's going to be a very interesting program, and I urge you to come and visit us. So thank you very much for your attention.
Thank you, Dr. German Serino, for your enlightening presentation and sharing your vast experience in the field of GM sugarcane crop. It is my privilege to introduce our next speaker, Padma Shri Dr. Bakshiram, the former Director, Sugarcane Breeding Institute, Coimbatore. The field of specialization is sugarcane breeding. In achievement, he had released so far 24 sugarcane varieties, and wonder variety developed by him is Co 0328, followed by Co 12029, Co 9004, and Co 11015. He had registered genetic stock, registered 14, and recently, he awarded the Padma Shri Award in the year 2023. He is RSA member for IISR Lucknow. He had, besides this Padma Shri Award, he had 25 different awards received. He has a vast experience of 40 years plus in sugarcane improvement. He had PhD guided PhD student 1 and MSc 1. Around 426 publications he had in international scientific journals, and total 36 book chapters he wrote, and database books 33. With this, I request Dr. Padma Shri Bakshiram sir to present on his topic breeding and contribution of Co 0238, a kernel wonder. Dr. Padma Shri Dr. Bakshiram sir.
Good afternoon. First of all, I thank VSI for inviting me to this conference. The chairman and the distinguished speakers, audience. I'll be, now I am a changed my field. Now I am an extension scientist, not a plant breeder. And I'll be, because so many young scientists may be sitting here, so I'll be what a technology developer should do extra other than the which is required scientifically or as per the job requirement to excel in their field. So my focus will be on those points. Of course, in India, sugarcane breeding initiated in 1912 with the establishment of sugarcane breeding center at Coimbatore. And the first variety was released CO205 during 1918, a cross between Saccharum officinarum and Saccharum spontaneum. And this was achieved under the leadership of Dr. C.A. Barber. Later on, Sir T.S. Venkatraman took over and he started tri-specific hybrids, that is involving Saccharum barbari also in the crossing program. So in India, present day varieties are tri-specific hybrids involving Saccharum officinarum, spontaneum and barbari. Of course, a few clones are having Saccharum robustum also subsequently. And since 1930 onward, this varietal development program in India has sustained the sugar industry. You can see this graph, both yield and sugar recovery, there is consistent improvement in both the characters, that is sugarcane yield productivity as well as the sugar recovery. And I have mentioned certain varieties also which are important for both sugar recovery as well as sugarcane productivity. So at different places of time, different varieties have contributed towards yield improvement and sugar recovery improvement in the country. So the 2020 figure, these 5 major varieties in the country. The most prominent variety was CO0238 with 54.9% area, CO86032 with 16.5% area, COM0265 with 6.9% area, CO0118 with 1.27%, 2.43% area, and COLK 94184 with 2.33% area. So these 5 varieties occupy more than 83% of the total area of sugarcane in the country. And the 22-23, the subtropical region, CO0238 continued to be the leading variety with 72.49% area, and during current season, area has gone down. It is still occupying 59.9% of the total sugarcane area in 5 subtropical states, that is Haryana, Punjab, Uttarakhand, UP and Bihar. And next variety is CO0118 with the 10.69% area, and during current season again it had increased to 16% of area. And COLK 94184 and CO80 98014 are the 2 other varieties. So if you see the contribution of kernel variety in the subtropical area, it is 85%. Approximately 85% area is covered by varieties developed from the kernel center. So coming to the wonder variety CO0238, in all the major states, apart from this variety crossed the boundaries of the zone and was cultivated in uh, Bihar, Odisha, and Madhya Pradesh and Gujarat also, and covered a more than 80% area in different states. So during 2014-15 to 2019-20, that is 6 years, the area of this variety during 1920 was 25.88 lakh hectare area, that is 2.59 million hectare area. And this is a rare combination which has increased. These two characters are negatively correlated in sugarcane, but this variety combines both yield and quality. So yield improvement of 5 subtropical states was 2.44%. This is unit improvement. And cane yield was improved by 19.7 tons per hectare, average productivity. If I have calculated the economics also, so additional benefit to the sugar factory as well as the farmers was 67,010 crore rupees from this particular variety. And additional benefit to the farmer was 53,000 per hectare. So there's a huge economics with this variety. If you see the improvement in yield and recovery during this period, during the period of this particular variety, say from 14-15 onward, there is a significant improvement in productivity as well as sugar recovery in the country. So if you see, divide the two parts, you see the lowest line, that is subtropical India, and 2013-14 is the base year. From 2014-15, the area started increasing at a very fast rate of this variety. And for average yield, subtropical India increased to more than 20 tons already I purchased, and it almost all the regions, this is national productivity as well as the rest of India, the productivity are at the same level during 18-19. If you see the sugar recovery also, the sugar recovery which was very low, less than 9% of subtropical region of the country during 2010-11, and from 13-14 onward with the increasing area of CO0238, it crossed the national average as well as the rest of the India average. And if we talk about the in UP, because UP is the major beneficiary from this variety, with the increasing area from 3% during 2013-14 to 86.7% during 2021, both yield and sugar recovery has improved. So more than 2.5% sugar recovery improvement, and yield also increased to more than 23 tons per hectare for the UP state. And of course, with the incidence of red rot, the average productivity is increasing, but recovery there is declining trend in the UP state now. So it has resulted in the increase in export of sugar from the country. You can see these figures from 2014-15 to 2021. The quantity has improved, the value has improved, and with the increasing area of 0.238. And this is the cumulative growth rate of sugar sector. In all the cases, the subtropical India, there is significant improvement in the cumulative growth for whether it is cane yield, sugar recovery, or sugar production, or sugarcane production. For all the 4 parameters, subtropical India is having higher cumulative growth rate compared to rest of the countries or India as average. And it has sustained the growth rate of the national level also because of improvement in the subtropical region. Then I have calculated rate of return on investment also. So this cross was made during 1996 crossing season. And 2009, this variety was released. So 13 years expenditure, if you see, of the Karnal Center, which was 27.63 crore rupees. And in 6 years, that is 2014 to 2019, it has earned 67,010 crore additional income to the farmers. So return on investment was 2,425 times of on the investments. And this particular variety has led to change in the policy decision of the government, because earlier B-heavy molasses and use of syrup, sugarcane juice was not permitted for converting into ethanol, but because of excess production in the country, these two decisions was taken by the central government, that permission to produce ethanol from B-heavy molasses and from sugarcane juice, and then rates of ethanol based on sugarcane juice, B-heavy molasses and C-heavy molasses. 3 different rates were announced by the government after this variety. There are so many socio-economic changes this variety has brought. Different people are telling so many things that everything increased in income of farmer, profitability of the farmers or the sugar factory has increased because of higher sugar recovery. Investment in agriculture sector has improved because farmers are having more money with them, available with them. Better education to the children, improved housing, and all these things I have seen at different places, particularly if we go to Eastern UP and Bihar. People are telling this has happened only because of cultivation of CO0238. So what next? Now this variety has come to red rot disease in the subtropical India. It started with Bihar and Eastern UP and spreading towards Western UP also this year. So area which was highest during 2021, 86.7% in UP, it has come down to 59.9% during the current season, that is 23-24. But another variety from Karnal Center itself is increasing at a very steady rate, that is CO0118. This year, current season, the area of this particular variety is 16.49%. It is slightly lower in cane yield compared to CO0238, but slightly better in the juice quality. But with the change in packaging practices of this variety, the yield has improved, equal to CO0238. So many other varieties have been released. This is the list of varieties released during last 8 years, released and notified in the country in different states. Almost 9 varieties per year are being have been released on an average during this particular area. So farmers are having enough choice to decide about the suitable variety which can replace CO0238. This another variety, CO15023823 from Karnal Center itself, having very potential. Of course, yield is slightly lesser, almost 5 tons per hectare. Yield difference is there compared to CO0238, but sugar recovery is higher compared to CO0238 in this particular variety. And this variety area of this variety is increasing in subtropical part of the country. This is another variety recently released, CO6030 from Karnal Center. Of course, mid-late varieties nowadays, area under mid-late variety is negligible, very less, because major area is under early maturing varieties. This is another variety, CO17018 from Karnal itself, yet to be notified, but have been identified for cultivation. Again, a mid-late variety And all these contributions has led to this award to me. Thank you very much.
Thank you, sir. Very nicely presented.
Thank you, Dr. Bakshiram, sir,
Very nicely.
for your inspiring presentation and sharing your experience with us today. I invite your attention to introduce our next speaker, Dr. G. Hemaprabha Madam. He is Director, Sugarcane Breeding Institute, Coimbatore. He had 37-year experience in genetic improvement of sugarcane by conventional and biotechnology. He had released 30 sugarcane varieties, registered 15 sugarcane varieties, and genetic stock registered by her is 18. He had awarded Sir T.S. Venkataraman Awards and NAS Fellowship. He had guided 4 PhD students or more than 300 publications on her name and 19 books he wrote. It is pride to tell me that she is the first woman director of the 112 years old Sugarcane Breeding Institute. So I request Madam to give her presentation on perspective of sugarcane breeding for value addition and product. Dr. Hemaprabha Madam, please.
Distinguished delegates on and off the dais. At the outset, I thank the organizers of VSI for providing me this opportunity to talk to you about the perspectives of sugarcane breeding and value addition and product diversification as it is happening particularly at India and very specifically at Sugarcane Breeding Institute. That is the action points on the issues facing sugarcane agriculture, particularly in India. So, around 120 countries grow sugarcane, and majorly it is cultivated in the countries like Brazil, India, Thailand, China, Pakistan, Mexico, Indonesia, etc. And coming to specifically the problems in India, that is area is around 5 million hectare, and slightly it is increasing now because of the bioethanol and other opportunities. And our problems, gray areas are mainly in the number of ratoon crops, which is very dismally poor, 1 to 2 ratoons. And the area under mechanical harvesting is very, very poor. Though, of course, with the availability, with the non-availability of labor, factories are moving towards mechanization, particularly very fast in the southern India. So some of the achievements, like about the varietal role, as just now you heard, one variety can change the entire scenario of sugarcane agriculture in the country. And the exports also started picking up from 2016, 2017, as the variety Co238 started spreading. And last year it was very high, around 112 lakh tons, so sugar was the highest agricultural commodity to be exported from India compared to other like milk, meat, or fish, or even rice. Sugar was the highest, led the commodity under export. And of course, about the bioethanol policy, which you heard from the main person itself, the horse's mouth itself, from our honorable minister. And there are so many products, like from food to feed to fertilizer to furniture to farming, that is even biofarming and producing value-added products. And even the edible and inflorescence of Saccharum edude is now is a preferred vegetable in many of the island, especially in the New Guinea and New Caledonian Islands. Now coming to the research priorities, now has the sustainable sugarcane production, as almost all the speakers spoke, and especially for India, because 238 made a revolution, and to sustain that sweet revolution is a very big challenge, and reducing the cost of cultivation, and make sugarcane agriculture more climate resilient and resource efficient, and product diversification. I will be speaking mainly on the breeding challenges, or the challenges how we can overcome through breeding. So first is breeding strategies to improve varieties for sugar, ethanol, dual purpose cane and size specific varieties so that the productivity can be improved. Then fast tracking and improving precision in breeding. Then good quality breeder seed, as everybody knows the importance of breeder seed, and the varieties for a targeted yield. That is, a variety alone cannot do. A combination of agronomic practices which can make that possible for an improved yield. And for that, it's our industry or in the institutes cannot do it. It is a collaborative approach between the industry and the research institutes. Now, there is a very unique system in India. Of course, all the research institutes have research priorities, but in India, the research, that is breeding, is likely centralized because there is a facility at Coimbatore and where all the parental clones are planted. And it's a national facility where 24 stations across the length and breadth of the country participate in breeding program. It's a very unique thing specific to India. Now coming to the breeding strategies, that breeding superior varieties for different maturity groups as well as location-specific varieties. Then enhancement of sugarcane germplasm, that is mainly to address climate resilience and resource use efficiency. And genomic and molecular approaches mainly to improve precision and improve selection. And the gene discovery, that is add a new trait, correct a deficiency in an otherwise promising variety. Then cropping system centered around appropriate varieties. Then enhancing the physiological efficiency as told by Dr. Prakash. Then host resistance, of course, is a dynamic thing because varieties break down to diseases and host resistance need to be improved. And economic and analysis to know both to address the gene-to interaction as well as the impact of varieties, how they are impacting the society, and of course the transfer of technology to the grassroots level. So there is no, I am skipping these slides because you know the importance of 0238. What made us here today probably because of the success of 238 and the higher income the factories and farmers are getting today. And there is no doubt that the person was awarded, that the breeder was, the scientist was conferred with the highest civilian award in the country. And there is another tropical variety, of course, that is 86032, the main variety in Maharashtra. We can see here, you are telling about 80 tons per hectare, 100 tons per hectare, 120 tons per hectare, 324 tons per hectare. Everything has been demonstrated not by one farmer, by thousands of farmers in the Sangli region in Maharashtra, where 325 tons per hectare is a very common thing, and even up to 167 tons per acre per, that is 18 months have been realized. That was the highest yield realized, that is coming around 425 tons. And this has already happened, not with one farmer, with many farmers in the state of Maharashtra. So that is the advantage of variety when it is coupled with suitable agronomic practices. Of course, everything should start with an improved high quality seed. Micropropagation with virus indexing will help you to get a clean seed, clean micropropagated seed, and using that in a breeder seed program. You can see in the background, this is a 7-month-old crop from tissue culture plants. So this not only assures a production of high quality breeder seed, but you can see how the seed farmer gets the yield. At 7 months, he is able to get 110 tons per hectare. So his profit, you can imagine the additional earning by these seed farmers. So not only the seed farmers are providing good quality seed, but his own income is also improving this way. So now next major thing is reducing the cost of cultivation. Among all the countries, the cost of cultivation is highest in India. So main strategies are to improve the multi-ratooning potential and also to develop short duration varieties mainly to realize 3 crops for 2 years or for a country sustaining the sugar recovery. And hybrid seed production through true seed. I'll be explaining in one or two slides that. Then mechanization, complete mechanization from seedbed to harvest. And of course, the doubling of farmers' income through integration of technologies. And here, the short duration varieties are very important. We have already released one short duration variety, which is as well climate resilient or waterlogging resistant, and which has a very high sucrose content. Of course, it has been released for the peninsular zone. and its suitability to other areas need to be worked out before the variety is spreading to the farmers. Because we get the information of this varietal spread, but it can lead to other problems. So the variety has to be tested in research stations. So now true seed, that is, that's a dream project of our former director, Dr. Bakshira. And that is the reducing the seed weight from truck to pocket. We need just 32 grams of seed for covering 1 hectare. That is how to make that seed possible. So we have tried several mechanisms, and in this one, the repeated selfing has been found to be very useful. Repeated selfing, identifying the selves, then molecular profiling, and advancing the best selves. And through a marker-resisted selection, you can go for selecting the best inbreds to make hybrids. And these are the hybrids at the 6th generation inbreds when we develop. We found a population which is near uniform, more than 90% uniform, showing that true seed-based agriculture is possible. Now we have to refine that technique to reach to the commercial level. We have also demonstrated that at the 7th generation, there is a high level of inbreeding depression. By crossing to 6th generation, hybrid vigor is obtained. In the case of sugarcane, hybrids look like very uniform and look like the normal sugarcane varieties. That shows that true seed in future that again requires more research to take a true seed in a pocket or in a packed cover and then develop seedlings, seedlings and plant in the field. So futuristically, that is another research we are strongly at. And then climate resilient sugarcane develop varieties, and also for resource use efficiency, not only salinity, waterlogging, drought, diseases, pests, of course for India, particularly red rot. And here there is a need for pre-breeding as well as speed breeding, because pre-breeding is a very slow exercise in sugarcane. It takes 30 years to realize a variety. So here it has to be combined with speed breeding, and of course gene identification and marker development. And for that, India has the highest germplasm collection. International collection is coming to 806 international collection. Apart from that, we made several collections through several expeditions, which is even now in vogue, collection of new types and under different categories of species, exotic hybrids. Then we have co-varieties, co-allied, interspecific hybrids. Then germplasm utilization line, cytoplasmic diverse line, everything is accounting to around 7,000 lines available at SBI. And here there are very specific and targeted pre-breeding work efforts going on, mainly to improve the species as such, that is intra-specific improvement of species. Then cytoplasmically diverse, because our hybrids are represented by the carum officinarum as the female. So we have a spontaneum as well as Erianthus as the female parent. Then again, hitherto unutilized typical officinarum to be mobilized in breeding, and race-specific and germplasm enhancement, mainly for specific characters like red rot and drought, and creation of that is magic population encompassing 8 different sources. And we have a very strong cytological analysis by which we ensure, because so far it is only the 2N64 types of spontaneum. We found that 2N90, 2N80, 72 are better than 2N64 in nobilization process. So the concentration is more on the high chromosome types of spontaneum. And also we gave more emphasis to the root mass, because whether it is for drought or for nutrient uptake or for mechanization, a strong root system is very much essential. So we have identified some of the spontaneum lines which have a very high root volume even under drought conditions, and there is very less biomass reduction under drought. These are being channelized into breeding program. And of course, another species that the Erianthus, these 2 species, namely Erianthus arundinaceus, known for its gigantic growth as well as multi-retooning efficiency and drought tolerance, and the Procerus types, which give very high proportion of red rot resistant parents, are now being utilized in breeding program. And in salinity studies with Erianthus have found that there are many types which record higher net photosynthesis, low canopy temperature, and significant high upregulation of saline stress tolerant genes. So Erianthus lines identified for high salinity are also used in breeding program, as well as in gene identification and used in genetic transformation. And GISH technique is helping us in identifying the true intergeneric hybrids, which are generally difficult to identify. These are with regard to using sugarcane with sorghum and sugarcane with maize. And we also identified several patterns of transmission of chromosome. There are 2n plus n plus n plus 2n, and different, and even androgenetic hybrids are also present. And there is a high level of pistoloidy representing, which limit the success of interspecific intergeneric hybridization. And also tri-specific hybrids are also included. And it's a success story that Arianthus procerus, we could get 2 co-varieties which have entered the ICRIP, including the new source of procerus, which is a non-cane forming type, but which give very high level of both drought tolerant as well as red rot resistant clones. And you can see there are many types within the R and MR category for red rot resistance. And cytologically also they have been characterized. And through molecular cytology, we have found that the BC2 clones having 5 chromosomes of Arianthus procerus, 5, 9, and 6 chromosomes of Arianthus procerus, showing that they are true hybrids. And also we are moving towards the chromosome additional lines, mainly with the procerus lines. Again, value addition in sugarcane, which nobody has thought of. We have red flush, that is robustum. After so many years, a few clones of sanguinium flowered. We made use of the opportunity, and now the BC2 hybrids are available, which are rich in antioxidants. That is sugarcane juice, all the goodness of sugarcane juice, plus the value of antioxidants. Now that work is now progressing for value addition in sugarcane. Genomic approaches, so many studies carried out, and several genes have been identified, and even marker-assisted selection. We expect that first the mass product will be available by 2026, and especially for drought tolerance. Red rot also, many markers have been identified from India and from elsewhere, from our work by Dr. Vishwanathan and others, and also from other group. So these are in the stage of validation, and our group is working on even doing marker-assisted selection for red rot. Then of course, genomic selection. This is the result of a collaborative program with Sugar Research Australia, and with high throughput phenotyping also in coupled with this genomic selection, we are able to make a headway in this line. And this is the principle. I am skipping this, but how a genomic selection model can work. And even for sucrose, we found that there is 81% prediction accuracy using selected genomic selection models. And this has been even tried for red rot. The 2 groups of linked markers have been identified in the same chromosomal region. And using these 2, the selection is around 72%. So this work is under validation before publishing. And of course, to aid all these things, we have to have a very strong speed breeding program. Of course, our team has standardized the protocols for inducing flowering, because unlike other crops like wheat and rice, where it is well standardized, sugarcane is a short day plant, and the totally different conditions were needed in the case of sugarcane. And of course, transgenic technology, as it was told in Brazil and Argentina, we have a system already in place from 1999. But again, the restrictions, of course, 17 events are now ready for limited field trials, especially for Co-86032 with enhanced drought tolerance, and again, and also Co-238. Dr. Apunu and team, Apunu is already here, and his team is working on that. And several physiological parameters have already been studied, and several genes have been identified from our germplasm, along with the suitable promoter, which have been used in genetic transformation experiments. Several transgenics, even single gene as well as pyramided lines are available, both through agrobacterium as well as the biolistic method. And of course, gene editing, it's like in very infancy in India. Of course, there are reports of the canacanes from Brazil. We are now identifying with a few traits, maybe 3-4 genes, and some events are available in the case of ILD. This is again, the team is sitting here, Dr. Apunu and Dr. Vishwanathan. Some lines are now available which need to be validated. They are in the B0 line. And of course, sugarcane as a bioenergy. And there are 4-5 strategies to improve the feedstock in biofuel. What we are mainly working on, it is lignin modification, that is altering the SG ratio And for that, already a gene has been identified, and that gene is being utilized. So that is a ferulate 5 gene is being modified. And now the energy canes is another Arianthus arundinaceous derived clone. This has already been now in the PPP mode, already gone for commercialization, mainly for fodder, biogas, hydrogen, paper, particle, and 2G. And this is the high biomass. You can see very high fiber as well as high biomass with this. And several, these have been commercialized. And biomass, and again the sugarcane as a platform for value-added molecules. And here the vacuole of sugarcane, where 90 to 95% any product can be targeted. We have a vacuolar targeting sequence, which is in the very final stage of patent. We'll be obtaining the patent very soon. So using that, our team is working on mainly for 3 genes, namely the glucose cerebrosidase, which is a rare genetic congenital disease, and also for insulin and interferon. And here this the downstream process standardization is in progress for the GSCS, and it's again within a PPP mode with a laboratory called Rana Lab. And several of our technologies for product diversification are available, like value-added products from sugarcane. And our entrepreneurs are being given hand-holding as well as training in this aspect. And now this is the last slide. That is now era of collaboration. Researchers have to collaborate with industry and vice versa, so that the expertise for the product delivery, refining, and value addition can reach the end users. And I thank also the ISMA for coming forward for a collaboration with SBI for a site-specific varietal identification on a pan-India basis. And with that, I thank everyone for this patient listening, and thanks for the opportunity. I also want to make an announcement that the 13th germplasm and breeding and the 10th molecular biology workshop of ISSCT will be held in India, where for the very first time since the inception of ISSCT, no conference is held in India. It's a rare opportunity. It will be held from 8th to 13th. It will be from 8th to 12th July 2024. It's a very rare opportunity. That's an international conference where many delegates will be coming from abroad. And this is the first time it will be hosted by Sugarcane Breeding Institute, Coimbatore, in collaboration with the industries. And I request all of you to be a partner, to come and participate in this one. Abstract has already been, submission is has already commenced. And 29th February is the last date. Dr. Butterfield, who is sitting here, is one of the co-organizers. And I again welcome you to SBI for this very first ISSCT plant breeding, that breeding and germplasm molecular biology workshop to be held in Coimbatore. Thank you very much.
Thank you, madam. Very nicely presented. And we explained all the perspectives of the sugarcane breeding for value addition and product diversification.
Thank you, Dr. Hemaprabha Madam, for your compelling and innovative presentation. It is my privilege to introduce our next speaker, Dr. Michael Butterfield. He is Data Science and Scientific Affairs Manager at CTC, Sao Paulo, Brazil. His field of specialization is plant biotechnology and breeding. He is leading a large team focused on developing a new sugarcane varieties with a higher productivity, adapted to the different agroclimatic zones and management practices. New technologies in support of the breeding programs, working on biotechnological tools to improve the productivity in sorghum, millets, and groundnut, chickpea, and pigeon pea. He is a major focus area. He chaired the Molecular Biology Committee in ISCT, and he also chaired the committee in germplasm and breeding in ISCT. With this, I request Dr. Michael Butterfield to give his lecture on the next century of sugarcane improvement. त्याचं काहीच उपयोग नाही ते ओपन करून. काय ओपन करून? त्यांचा बायोडाटा दिसला पाहिजे मी सांगतो.
Good afternoon, everyone. First of all, I'd like to thank the organizers, VSI, for inviting me here to present today. It's a wonderful opportunity to be here with you all. So for me, context is very important to understand where we are and where we are going, and also to understand what opportunities there are. So what I intend to do today is to give a little bit of context regarding sugarcane productivity, to see what that means when we think about the future, especially when we think about in terms of sustainability. So as we've already heard today a few times, sugarcane is one of the most efficient plants at fixing biomass through photosynthesis. And there's been various studies to try and estimate the theoretical limit of productivity. And one of them that has been quite widely quoted is this theoretical limit of about 380 tons of cane per hectare. And if you take an average over the year, about 13.5% sucrose content, that would translate to about 50 tons of sucrose per hectare. So if we think about modern sugarcane breeding, it started about 100 years ago, more or less. So if we think about, as we heard today already, Code 205 was released in, 1918, Code 213, 1928, POJ 2878, 1921. And also the Hawaiian crossing solution was developed in 1924. So that's its 100th anniversary this year. So if we look at the progress of the last 100 years, what does that tell us about the next 100 years? This graph here is showing the yield progression in Brazil over the last 100 years. So going back into the early 1900s, the yield of sugar, and here I'm presenting data in terms of tons of sugar, not tons of sugar cane. So back in the early 1900s, Brazil was producing about 2 tons of sugar per hectare. And then really about in the 1960s, with new technologies, breeding, etc., that increased to a maximum of 12 tons per hectare. So in fact, over the past 100 years in Brazil, the sugarcane yields increased by a factor of 6 times. And that's really due to the combination of improved varieties and also coupled with management. So better fertilization, spacing, irrigation, weed and pest control, etc. But if we look at the last 20 years in Brazil, the yields have basically plateaued at about 10 tons per hectare. Part of that was due to the introduction of mechanization in Brazil, where you have additional losses during the harvesting process, and then also damage to the stools and the ratoons. So although the yields have increased impressively by 6 times over the last 100 years, over the past 20 years, the yields have been more or less static. But what does this mean when we think about what the potential of sugarcane is, and what we can do to try and reach that potential of 50 tons of sucrose? So as I mentioned, in Brazil at the moment, the average is about 10 tons of the sucrose per hectare. If we think that the theoretical average is about 50 tons, that means we have a 40-ton gap in terms of yield. And so this 40 tons gap, is this due to genetics? Is it due to environment, management, and what can we do to try and fix this? So although the average yield in Brazil is about 10 tons of sucrose, as we've seen in some of the earlier presentations today, in individual fields, the yields are much higher than that. So in Brazil, at CTC, we keep a big database of commercial fields over many years. So we have data on more than 19 million hectares over 10 years. And if you look at the top 10, the top 100 fields of that, the average of those top 100 fields is more than 40 tons of sucrose per hectare. And if you look at the top 1%, it's about 30 tons. And we can see reports from other industries as well. So in Australia as well, there's commercial fields that are producing more than 30 tons. And we also see in India some examples of very high production. So what that means is that the average production is 10 tons per hectare. The actual genetic value of our current germplasm is in the region of 30 to 40 tons per hectare. So that's already 70 to 80% of the theoretical limit if we take that as 50 tons of sucrose per hectare. So what that means is that yield gap of 40 tons is made up about 15 tons of genetic gap, which is about 40%, and about 25 tons of environment/management gap, about 60%. So what can we do to try and close this gap? So obviously breeding will play an important part, but I will come back to this a little bit later. But as we've seen in the previous slide, management, but management in its broader sense, including environment, etc., has even a bigger role to play. And without this, the genetic potential of our current germplasm can't be attained. And especially when we think about the future and climate change, managing for resilience will be really key for sustainability. Because as we've seen from some of the presentations this morning, climate change means more extreme events. So either more drought events or more flood events, but that can happen in the same region. So it's one thing to breed varieties for either drier conditions or wetter conditions, but it's very difficult to select varieties that will perform well in both dry and wet environments. That's why resilience is really the key when we think about management and environments. So when thinking about climate resilience strategies, there's a whole range of things that I think the industry needs to start taking cognizance of and looking at seriously. So this is just a summary of some points that were raised in a review published in the ISSCT latest bulletin from December. So things like soil health, as we know, increasing organic matter in the soil can increase water holding capacity, which has drought alleviating effects. Doing structural things to help with rainwater harvesting, reducing runoff, water storage, water efficient irrigation, precision agriculture, as we've heard today, with drones using nano formulations, etc., and also coupled with weather forecasting. Agroforestry integration, this can help also with extreme weather events, thinking of wind and lodging, and also variety selection. So in terms of variety and selection and breeding, as I tried to show in the earlier slide, the breeding has already pushed the genetic potential of our varieties to about 70 to 80% of the theoretical limit, which makes it even harder to push genetic gains by breeding further. But really the first thing is to get the basics right in terms of breeding. So you need to select the best parents, plan the best crosses, select in the right target environments, and also phenotype appropriately. And only when you have those basics well established that it's worthwhile thinking of going to modern breeding techniques. And when we talk about modern breeding techniques, I've used the slide here from a publication of a former colleague of mine at ICRISAT, Rajiv Varshni, where they talk about the 5 Gs for crop improvement. So the first G is the genome. So we need to have genome assemblies for our crop species, both sugarcane and also the wild species. We need to have the germplasm well characterized at both the genome and the agronomic level to be able to take advantage of these genome assemblies. We need to have, based on the genome assemblies and the characterization, we need to be able to identify genes and also functionally characterize them. And using that, we can then start using genomic breeding approaches such as genomic selection. And then finally, we can then also, with that information about gene function, we can start using gene editing to improve genetic performance. So just in terms of where we are with these 5 Gs approaches at CTC, so there are 5 public saccharum genomes available in the literature, and also we have additional CTC genomic information. In terms of germplasm characterization, we have over 9,000 clones, which have been well characterized for many traits, and also each of those clones have been characterized with more than 50,000 SNP markers. In terms of gene function, we have transcriptomes of 2 CTC varieties with gene adaptations, and also there's many public information available on NCBI. In terms of genomic breeding, based on the germplasm characterization and the SNP markers we have genomic selection models to select both for sucrose content and for sucrose yield in different environments. And also in our laboratory in St. Louis in the USA, we have various proof of concept projects using gene editing, using some of the information that we've gleaned from the genome and the germplasm approaches. But it's all very well having huge quantities of data, but often the implementation of that is not trivial. So this is just another example of a publication, also from Rajiv Varshni and colleagues, looking at grain yield and rice. So in this project, they identified 120 relevant genes. And there on the top part of the figure, you can see all the genes listed and what function they have in various parts of the phenotype. And based on characterization of the germplasm, they were able to identify 21 specific haplotypes or gene variants amongst these 120 relevant genes that really have the greatest impact on yield. But it's still a great challenge to assemble these 21 haplotypes into one genotype to actually realize that yield. And in sugarcane, this is even a more difficult thing to accomplish, seeing that we don't have back crossing to integrate those genes. Thank you. And also to think about how you would apply this in sugarcane. And obviously drought is something that everyone always talks about, but as Prakash already mentioned, drought is an extremely complex phenomenon with a long list of phenotypes that contribute or potentially contribute to drought. But you would have to do this kind of exercise looking at the genes involved, the haplotypes, etc., which is a very long and complex process. So just to summarize, if we assume that the theoretical yield limit for sugarcane is about 50 tons of sucrose per hectare, Modern varieties that have been developed over the last 100 years already deliver 70 to 80% of that yield potential when they're grown under ideal conditions. As conditions change, particularly with climate change, these new 5G tools will be needed to complement traditional breeding, to maintain these yield gains and to close the yield gap. But in addition to the breeding, adaptive management strategies will be essential to realize the yield potential already provided by the improved varieties. Because without this, there's a high risk that sugarcane production could be negatively impacted by climate change. Thank you.
Thank you, Dr. Michael Butterfield, for your insightful and inspiring presentation. With this, we had completed the 5 presentations, and with this, I hand over to Honorable Chairman for further discussion.
Now, all speaker has made their presentations now. Very nicely they presented. Any question in mind? It is a time to question. They can speaker can give the answer of your question or satisfy you. Please, yes, please give the mic to him.
Hello.
Thank you all the speakers for fascinating and riveting presentations. There are lots of questions to ask, but I'll only ask one. I hope what I'm going to ask is not a frivolous question, but in the opening presentation by Prakash, you know, as he rightly pointed out, it is concerning that only 8 cane cultivars dominate the global sugarcane production. So what I really want to ask you is, with the current advances in synthetic biology, what is stopping your alter ego to exploit mRNA technology for bioterrorism purposes against those 8 cultivars, and therefore make more money on the futures market than being a researcher?
Thanks, Aravind. It's a good question. Now, mRNA, the biotechnology, various aspects of biotechnology has been tested in sugarcane in so many traits, and under experimental conditions, a lot of traits did really well as expected. But from my experience, I myself, during my career, tested at least half a dozen different traits. When it is taken, the material which are considered to be quite superior to the field, taken to the field, all those wonderful results I have seen in a controlled condition disappear. So, like what I was telling through my presentation, and at the last presentation, Mike was saying, it is not pushing more on the genetics. Genetics already really has a huge potential. But what is happening is the genetics we have, the varieties we have, are not really expressing its full potential. So my wisdom coming from all my experience is that probably you need to spend more time and effort and thinking and innovation in getting the best out of the current genetics to minimize the yield gap. That is what going to be making big quantum leaps in sugar production in the coming future. However, a few technologies like a more detailed characterization of genetics or genes in relation to function, which we can do it now, which was not possible 10, 15 years ago. So some very targeted effort, which if you have really a good understanding about how those genes function, and then exploiting the new biotechnology tools to make use of those genes profitably will make sense. But again, the varieties and its genetic potential is not the one holding us back what we can't realize the real potential out of those varieties.
Thank you, Dr. Prakash. Next, anybody who doubt in mind, please give mic to him. Yeah. Please come forward.
Hello, I'm Sendil. Hello,
Yeah.
I'm from Hyderabad. Actually, my question is on this transgenic cane. Actually, Mr. Serino, he was indicated it was approved and cultivated in Argentina. What is the status that?
Please come forward, come here, and because we are not understanding your voice.
It is a question on transgenic cane. So during the lecture of Dr. Serino, he indicated that it is approved and cultivated in Argentina. What is the status in Brazil and also in Thailand? And also during the lecture of Director SBI, she has indicated the status of study on transgenic cane in India. So when we can get transgenic cane in India?
Yeah.
First status on Brazil and Thailand.
Variety is approved.
Yeah.
Yes, can you help me? I'm just having too much noise here. I'm asking them to explain the question.
So if I understand correctly, the question is about the transgenic sugarcane is already in commercial production in some countries like Brazil and in Indonesia. And what is the prospect of transgenic material, transgenic sugarcane for the Indian production? Is that what is the, is it?
Sir, what is the status in Brazil and also Thailand? Are they having transgenic cane in their countries?
It's, transgenics are widely grown in America, in Southern American countries, and it is banned in Europe. India is having a medium stand. Now GM cotton, it is approved.
Yes.
But food crops are not approved in India because of biosafety regulations and the agony or some questions raised by the environmentalists. With that, the transgenics are not allowed to be released in India unless they are proved to be very safe. BT cotton is released, BT brinjal is hanging, BT mustard, it is just on the anvil that the government has given 2 more years for further studying before it is released. For us, 1999 transgenics have been standardized, the technology has been standardized. We are ready with the drought tolerance, increased drought tolerance in 86032. We are waiting for the clearance from the government because in India, there are 4 stages of getting approval. Now we have approached RCGM to give clearance for limited field trials. Once the limited field trials are okay, then we have to go for Ministry of Environment and Ministry of Agriculture, and then only the variety can be released. But the problem is that because it has a foreign protein, I'm not explaining all these things. It's all governed by government policies. So once the government is ready, we are always ready. Research is always on that angle. Now that because unnecessarily we are working and producing more plants, now we have just going slow. All the events are maintained in the glass houses. once we get approval, GM sugarcane with improved drought tolerance will be in your hands.
Yes,
But
thank
in
you,
other
madam.
countries, there is no issue. 70,000 hectares in Argentina, in Brazil still more.
Yes.
So maybe I can answer your question about Brazil. So in Brazil, we have 4 transgenic varieties that are released for commercial cultivation. The first one was released in 2017, and since then we have had 3 more releases. Currently, I think there's about 40,000 hectares planted with those GM varieties. They are all resistant to insect borers, so they have the BT gene. And what we have been able to show, so although sugarcane is a food crop, we can show that in sugar is a purified substance, and ethanol is also a purified substance. So in sugar and ethanol, you cannot detect protein
Yeah.
or the gene itself. So we already have approval from the USA, from Canada, and also from China that they can receive sugar and ethanol from Brazil because it's not considered transgenic, even though that the plant in the field is transgenic.
Yes. Thank you. Thank you. One more question. In India, the popular varieties, you take CO238 is infected by red rot, and also number 2, 86032 with YLD and also Boca Boeing, and also in the case of 8005, now it is seriously affected by rust and also smut. Under this situation, how to manage, and also what is the recommendation?
238, of course, farmers are not willing to part with their variety.
Yes.
To some extent, we are managing through the clean seed program. And you know about our STD, that is a set treatment device. So through that, the delivery of chemicals, and it is being done in a very effective way to some extent. But in the case of smut, actually STD combined with hot water, I think Dr. Vishwanathan is here, is a remedy for that. Of course, breeding has to continue, and disease resistance breeding has to continue. But this type of minor diseases can be easily managed. YLD can be very easily managed through a virus indexing, tissue culture, the micropropagation method. You have recreated, or as a rebirth has been given to 86032. Now what is happening to 86032? Even in Maharashtra, the variety is now spreading. Area under 80s, that was 16.5% in 2021. Now it has come to 19%. Area under 86032 is improving or increasing because tissue culture, micropropagation has come to a rescue, and the original vigor has been restored. But other diseases, of course, because disease resistance is a continuous process, breeding for disease resistance, we have to continue. But we can manage some of the diseases effectively through good agronomic practices, especially through the STD.
Yes, thank you, madam. Thank you. We are also following TC in a large way.
Okay, thank you.
Sir, can development of CMS lines in sugarcane will commercially viable on large scale cultivation in India?
Repeat again.
Can development of CMS lines in sugarcane will commercially viable on large scale cultivation in India?
CMS line. CMS line is viable for large cultivation.
Will it possible? Development of CMS lines?
There is no need of CMS line because for you the commercial product is cane. CMS lines are come into picture when seed is the product. But there is no need of CMS line in sugarcane.
We have seen
Because it is cane is the stem is the commercial part, not the seed. CMS is cytoplasmic sterile line that will not lead to seed set.
But will it possible?
We don't want seed set in sugarcane. No?
But we can get more number
Not
of
need.
possible. That was my question. We can get more number of seed if possible.
Yeah, as per her opinion, this is not possible. Okay, thank you.
हेलो सर, हिंदी में बोल, आप समझ के बता देंगे जी? मैं हिंदी में बोल रहा हूँ सर। सर एक क्वेश्चन है, मैंने जितना भी देखा सुबह से, जितनी भी कंट्री के सभी साइंटिस्ट लोग या जितने भी आए हुए हैं, मैंने देखा कि सभी जगह गन्ने पिछले 2-3 साल के अंदर रिवर्स आया है, बढ़ा नहीं है जो शुगर केन की बढ़ना चाहिए। तो अपना ये है कि उसका रीजन तो समझ में आया कि क्लाइमेटेड इफेक्टेड सबसे बड़ी चीज हो रही है। तो हम ये चाह रहे थे कि उस पर क्या कर पाए कि रिवर्स ना होके गन्ने की तरफ ज्यादा लोग दोबारा वो होए, ये हम समझना चाह रहे थे।
जहाँ भी गन्ने का एरिया कम हुआ है वो ड्राउट के कारण हुआ। जो मॉनसून फेलियर मतलब रेनफॉल लेस हुआ है उसके कारण है, और कुछ नहीं। जैसे ही रेनफॉल बढ़ेगा तो गन्ना भी बढ़ जाएगा। महाराष्ट्र और कर्नाटका में भी इस साल कम हो गया है एरिया, वो ड्राउट के कारण हुआ है। जैसे ही एरिया बढ़ेगा तो इस साल अच्छी बारिश हुई, अगले साल बढ़ जाएगा।
सर हर साल जो क्लाइमेटिक कंडीशंस चेंज हो रहे हैं, तो क्लाइमेटिक कंडीशंस की वजह से जो गन्ने की यील्ड के ऊपर जो प्रभाव हो रहा है, तो उसको मेंटेन करने के लिए वैराइटल प्लानिंग हो या कुछ पैकेज ऑफ प्रैक्टिस हो जो खेत में करते हैं, तो उसके लिए क्या प्लानिंग रहेगा? ऐसा
देखिए, क्लाइमेट जो दूर की बात है वो तो रिसर्च वर्क चल रहा है उन सभी पर उसको देखते हुए, परंतु जो ऑल ऑफ ए सडन होता है उसके ऊपर तो मैनेज करना पड़ेगा उसी दिन में। अब जैसे मान लीजिए ड्राउट टॉलरेंस है, ड्राउट टॉलरेंस का हमारे पास बहुत सी वैरायटी है जो कि ड्राउट टॉलरेंस है, परंतु ड्राउट आता है कभी कभी आता है और वो अनप्रिडिक्टेबल है। महाराष्ट्र में आ गया, कर्नाटक में आ गया, कभी नॉर्थ इंडिया में आ गया, पता नहीं कब आएगा। परंतु जो ड्राउट टॉलरेंट वैरायटी है, सर्टनली उनकी यील्ड जो है नॉर्मल ईयर के अंदर कम होगी, उस चीज के लिए कोई तैयार नहीं है। हालांकि महाराष्ट्र के अंदर बहुत फ्रीक्वेंटली ड्राउट आते हैं। हमने VSI के साथ मिलके SBI ने ट्रायल कंडक्ट किए महाराष्ट्र के अंदर और 2 वैरायटी आइडेंटिफाई किया जिसमें कि वाटर ड्राउट टॉलरेंस था। 86032 से बेटर यील्ड थी उसकी, बहुत रिकवरी भी ज्यादा थी, यील्ड भी ज्यादा थी, परंतु रिलीज नहीं कर पाए क्योंकि अगले साल अच्छी बारिश होगी, एरिया बढ़ गया, वो ड्राउट वाले को सब भूल गए। तो ये इश्यू है और कोई नहीं है। और जो दूर के जो चीजें दिखाई दे रही है उसके ऊपर तो हर एक सेंटर काम करना शुरू कर रहा है।
सर अभी जैसे कि मध्य प्रदेश में लास्ट ईयर के कंपैरिजन में R1 और R2 का जो गन्ना कट गया है, अभी 1.5 महीने से सभी फैक्ट्रियां चल रही है, तो 4 से 5 टन का अभी जो डिफरेंस दिख रहा है, मतलब 4 से 5 टन के यील्ड कम आ रही है गए साल के कंपैरिजन में, और जो कि सभी शुगर फैक्ट्रियां बारिश के कारण 10 से 15 दिन लेट भी हो गई, लेट चालू हो गई।
Actually, now this is agriculture is fast changing. Now the era of precision farming, precision agriculture is coming. So that is mainly with the big data analysis as well as the artificial intelligence. So you can forecast how your yield will be based on the data from the previous 10 years, 100 years, or whatever it is. So based on that, it will be very easy to predict what the climate will be next year, weather will be next year, and accordingly, farmers will be advised to give the necessary agronomic practices to be followed for achieving the maximum or maximizing the yield. So that is coming in a very fast way. Probably in the next conference, you can hear more on the practical application of AI in solving the problems of similar problems. Now we are in a very early stage to comment on that, but definitely that is coming in a very big way. So future it will be very bright, and now we have to manage with the short-term goals or short-term weather data we are getting, we have to manage.
We are also planning for the same, madam. Thank you.
Okay, madam. Thank you.
Okay, thank you. Then you ask this question during dinner time, please. We will discuss afterward. Okay.
Smart, sir. So my question is to Madam. So this is regarding true seed production in case of sugarcane. So we know already in case of potato, it is not working properly. Whether it is going to be successful story in case of sugarcane, because you told that small quantity of seed is required, but for India, we know the area is more, and seed setting is, I think, mostly in case of Coimbatore area only.
To be very frank, we tried many approaches. Inbreeding, apomixis, wide hybridization, anther culture, many other approaches were tried. But finally, we have come down to inbreeding. That is, at the 7th or 8th or 9th generation, we are able to, that inbred into inbred, you could get higher yielding hybrid varieties. But that has to be replicated in a large scale to the latest variety. Now we are standardizing with the available inbreds. Of course, the development of inbreds will take a long time. So probably with the speed breeding facility which we are going to implement, it may be faster. Our target is 2050. Hopefully, if things are going in the right track, by 2035, true seed-based planting can happen, at least in limited area.
Thank you, madam.
Thank you.
Good evening to all of you, friends. Today now we are concluding now. Now 5 presentations made by different speakers, and very nicely they presented their presentations, and so many from house, so many queries has asked for their some doubt in mind. They also clarified this doubt also, and I appreciate and congratulate to all speaker for within stipulated time they cover this session. I thanks to all of them for giving me cooperation, and before 6 we are completing now the session now as per the schedule, and also our organizer has given me opportunity to share this session. Thanks for organizer to give me opportunity for this session, and this is a very unique experience for me for chairing of this such type of special session. And I congratulate to VSI organize this national conference, international conference here. Once in a 3 years, they have been organizing this conference. And previously, I also attended this conference, and now today also I am attended this conference. And very good response for that. Honorable Gadkari, sir, has given the very good speech about it, and they assure about the sustainable sugarcane production and also sustainable marketing in future. Thank you very much.
A sweet ceremony to honor today's honorable chairman and distinguished speakers. In this, I request Dr. A.D. Kadlak, Principal Scientist, Crop Production and Protection, Vasantdada Sugar Institute, to come forward and honor today's chairman, Dr. S.R. Gadak, sir. Sir, thank you, sir. I request Honorable Chairman Dr. Gadak, sir, to honor Dr. Prakash Lakshman, sir. I request Chairman to honor Dr. German Serino. I request Chairman to honor Padma Shri Dr. Bakshiram, sir. I request Chairman to honor Dr. G. Hemaprabha Madam. I request Chairman to honor Dr. Michael Butterfield. Thank you, sir. Now, before closing the session first, once again, I am grateful to all delegates who participated during this session and our honorable chairman and distinguished speakers who delivered the innovative presentations in this session and make this session a great success. I thank you, my colleague, Rapporteur Dr. Devarmat sir, for recording the deliberation. And once on all, I thank you all for attending this. Thank you very much.